Hi guys, I'm going to be talking a little bit about standard assembly as practiced in the Anderson lab. So we have a series of standard plasmids, standard vector backbones that make cloning really easy. So when we're talking about cloning, we do a lot of cloning in this lab. So we might have parts A, B, C, D, and we want them to go together. So we want to make, A, B, C, D, then A, B, C, D, and that might, that's an example. We could have E, F, G, and then we could have prime, or I guess, C, C1 through C, I don't know, 5. So this can complicate things, which would make this C1, CD1 through CD5. So I guess that's all besides the point, but this assembly tree, there are many ways to do this assembly tree, and I'm going to be talking about how we're doing it. So in our lab, we have a series of these standard plasmids. So I'm going to just draw you out one of these plasmids. Nice circle. Oh wow, that started out nicer. So here's our plasmid, and we'll say that this plasmid has part A in it. Now, this part can be whatever, but there's a few rules for this part. This part cannot have an ecosite, eco R1. It cannot have a bagel site, a bagel 2 site. It cannot have a BAM site, BAM H1. And it cannot have a ZO site. These are the off-limits enzymes. So no, don't use these enzymes. And if you have a part that you need, but it has, some of these restriction sites in your part, then you need to use a technique like sewing PCR in order to remove it. So for reference, you can check out the sewing tutorial to figure out how to do that. So we're going to assume that you've already removed all of the restriction sites from this part. The reason we don't use any of these we can't have any of these restriction sites in this part is because these restriction sites are going to be very important for our assembly. So let's take a look at our plasmid. We have a few landmarks. We have an eco R1 site. We have a bagel 2 site, bagel 2. We have a BAM site, BAM H1. And then we have a ZO site somewhere down here. In between our eco and bagel site, we have an ATG. And after our BAM site, we have a TAA. So start codon and stop codon. So we also have a few primers sites. We have a CA998, CA998 primer. That's a forward primer. And then we have a reverse primer. And this is not to scale. This is a G00101 primer. So this is our plasmid, and this is an awesome plasmid. We have a bunch of different flavors of this. Pretty much from here to there is standard with a few differences. And then we have different versions of our Ori of replication. So we have, Puck, we have R6K, we have Cali2, Cali1, so a bunch of different origins. And then we have a bunch of different antibiotic resistances. So resistance, so we have AMP, chloramphenicol, kanamycin, spec, trimethoprim, TET. So there's just a bunch of different flavors that you can use. And I just want to talk about a few more of these landmarks. So we have EcoBagel, BAM, and these two primers. So you might have noticed in some of my other tutorials that I always talk about whenever I want to bring up an enzyme, I'm always bringing up Eco or Bagel or BAM whenever I'm PCRing up a basic part. That's because those are the enzymes or the restriction sites that we're going to be adding on to A in order to get in this plasmid. So let's just say that we have A on the genome somewhere. Let's say that we're starting off with A, we have some genomic DNA, and A is over here. And we'll just pretend that there's no restriction sites, that we got lucky. If there were, you'd have to sew it out. And we want to get A into that plasmid. First thing we're going to do is design a few oligos. We're going to have an oligo which is going to be binding to A, a forward oligo and a reverse oligo. They're going to be binding to A there. And we're going to be adding on a little floppy tail to our oligo. And on this floppy tail, we're going to actually just put in this sequence. We're going to have a 5' spacer, so it's going to be, let's see, we're going to have our Eco, our ATG, our Bagel 2. That's Eco R1, to be clear. And then a 5' spacer. And that 5' spacer, allows us to digest. If we didn't have any 5' spacer there, like just 5 random base pairs of DNA, our EcoR1 enzyme will not be able to bind to and cut that DNA. So EcoR1 is an endonuclease, meaning it cuts within the DNA, versus an exonuclease, which means that it would chew back the DNA. So we need to make this restriction site internal. So we now have this Eco ATG bagel with a 5' spacer for a forward oligo. And then we're going to do something similar for a reverse oligo. We're going to have BAM H1, so that's BAM. And then a 5' spacer as well. So 5, oh, I have really bad handwriting. So that's a 5' spacer. We PCR that up, and we get a part that looks like A. And let me just switch colors again. So now we have Eco, Bagel, BAM, and our 5' spacers on each side. So don't forget about that. We have our 5' spacers. That's real important. Then all we got to do is digest with Eco and Bagel. So digest Eco and Bagel. Or I'm sorry, not Eco and Bagel, Eco and BAM. So Eco BAM. We say in our lab we Eco BAM it. So we're going to cut there and cut there. And we're also going to be cutting our vector backbone with Eco and BAM. So let's draw a new vector backbone. So we have Eco, Bagel, BAM. So E, BG, BM. And we have some sort of a dropout. Usually we'll just stuff an RFP in here or something like that, just as a placeholder, so that when you digest and run it on a gel, you can see that there's been a change in size of the two bands. So you have a dropout. So you should get any singly cut should be apparent. This is about 1 kb. And also an RFP will show up the next day as red and will tell you what clones are background. We've actually been using a slightly larger part called violocine, which is a I think it's a 7 kb part. And that just shows color much quicker, and it makes the plasmid the dropout, the vector backbone the dropout, instead of vice versa, so it's just easier to purify. Anyways, tangent. So we have our plasmid. We have our ORI. We'll just say that this is a puck. and then we have our antibiotic resistance. We'll say it's spec, and this combo is actually 1256 in our lab. So this is our plasmid. So we have a part, we've digested that with eco and BAM, we PCR'd it up, eco BAM'd it, purified it, cleaned it up, and then we're going to do the same thing to our plasmid. We're going to cut with eco, we're going to cut with BAM, and we're going to run it on a gel. So we're actually going to run both of these on a gel. We have our ladder, which is just going to be kind of telling us how big stuff is. The part we'll put right there. Our part should have just one band and it's digested. And then our vector backbone is going to have two parts. We're going to have the vector backbone, if we're using the RFP, which is 1 kb. So we're not using that cool part. RFP is 1 kb. RFP is going to run down here, and our vector backbone, which is 2.5 kb, is going to run up here. So then we have this gel, we cut these fragments out, we can then gel purify them. There's a tutorial on how to do that, and we can clean them up, and then we just end up with just this chunk, so none of that extra stuff, but just what is inside of this circle, or actually, wrong circle, let me change colors. So after gel purification, we end up with just what's inside of that green circle. Bad contrast, let's try blue. Just that, that's a little bit better. And for our plasmid, we're going to be getting rid of this. So we're going to be just left with everything else, everything outside. Just if I was to, it's going to be hard to draw a circle around it, but everything outside of that scratched out area. And then we have an eco site right here, which is compatible with that eco site, and a BAM site right here, which is compatible with that BAM site, so that when we put this part, this basic part, in with this vector backbone, we can ligate them. We just add some ligase. Again, we have a liquid handling tutorial on that. And you get this plasmid that looks like what we had originally. We get something up here. So our A just drops in at this eco and this BAM site. And that's pretty much how you do an eco-BAM transfer. And this is kind of the first step of an assembly. So then you would just transform that. You would grow it, you'd get colonies on a plate, so your plasmid then goes into bacteria through a transformation. Your bacteria go onto a plate, a petri dish, and you get a bunch of colonies. You pick those colonies and they go into culture tubes. And you grow those up in liquid culture overnight and you shake them. You can then, okay, I'm running out of space here. Let's see here. You can then mini prep. You'll purify the DNA and then you'll just get a ton of this. So I guess that would be kind of the cycle. So you get a ton of this, and then once you get a ton of this, you then do the QC on it. So this is QC. And the QC means you're just going to map it. And how we map it, we digest it with eco and BAM again, and we just run it on a gel, and we should see something that is the size of whatever A is, and then the vector backbone, which is 2.5. So if we run this on a gel, so the QC is going to be running an analytical gel, which is, you know, going to look similar to this. We should see our 2.5 base pair, 2.5 kb vector backbone, as well as however big the part is. So I drew this one right next door, so I guess the part would be something like right around there. After you run that on a gel, you can send it off for sequencing, and we use the primers CA998 and G001 for reads. If this is a small part, less than 700 base pairs, CA998 will be sufficient. If it's any bigger, you're going to want both a forward and a reverse read on it in order to try to sequence all the way through it. And that would be the complete QC cycle. Then once you've QC'd it, you're just gonna make sure that it's well annotated. You have a tube that it's very well documented that has been QC'd, and you have a record of that QC. Then you can go into the assembly, and we're gonna talk about how we assemble basic parts next. So if you have any questions on EcoBAM transfers and then QCing, just let me know, and we'll be back in a couple seconds.